Assalamu alaikum. I am Baina Saeed al-Marriya, student from Al-Arqam Academy Year 10, and this is my project, Novel Electrospun Nano Fibrous Scaffolds for Tissue Engineering Applications, and I worked on this project with professors from Qatar University's Young Scientist Center. Tissue engineering is an innovative approach aiming to regenerate damaged or lost tissues and organs. Scaffolds are used to fill damaged sites and finally form a functional tissue. Fenugreek is a medicinal herb rich in antioxidants which play an important role in wound healing. In this work, we propose the preparation of PLA fenugreek nanofiber scaffolds by electrospinning technique, which have shown very promising results in wound healing and of good mechanical strength and biodegradability. Wound healing consists of three main phases: inflammation phase, cellular proliferation phase, remodeling phase. We are aiming to accelerate the wound healing process using scaffolds and tissue engineering. For the materials we use, dimethylformamide, polylactic acid, dichloromethane. Our medicinal herb was fenugreek, and we also used trypsin and Delbuca-modified eagle media for cell migration, and phosphate buffer saline for testing biodegradability. For the equipment we used, magnetic stirrer, electrospinning machine, centrifuge, safety cabinet, cell incubator, scanning electron microscope, and tensile testing machine. Step 1. I measured out the solution, which is composed of DMF and DCM to equal 20 milliliters. This step was repeated twice for the PLA scaffold and the PLA fenugreek scaffold. Step 2. I weighed out the PLA polylactic acid. Step was repeated twice for PLA fenugreek scaffold and PLA scaffold. In the fenugreek scaffold, the fenugreek was also weighed out. Step 3. The PLA solution was mixed in the magnetic stirrer and left for 24 hours. Again, this step was repeated twice for PLA fenugreek scaffold and PLA scaffold. Step 4. The last step in the making of the scaffolds. The final solution was taken and put in the syringe in the electrospinning machine to form the mats. And as you can see in the following video, threads that form the mats. Experimental procedure for cell migration. On a 6-well plate, endothelial cells were grown. 2 PLA control, 2 PLA, 2 PLA fenugreek. The plates were supplemented with DMEM cell media. Scratch was made on the surface using a pipette tip to mimic injury. The control was left empty, PLA fenugreek scaffold on 2 cells and PLA scaffold on 2 cells. Results were observed in 72 hours. One, SEM analysis. Using Canon electron microscope analysis, it proved that the scaffold made was able to mimic in shape the tissue structure, as shown in figures A and B. 2. Biodegradability. The PBS was used to test the biodegradability of PLA scaffold and PLA fenugreek scaffold. They were left in the solution for 48 hours, and as you can see in the graph, as time passed, both of them lost weight, which indicates that both scaffolds are biodegradable, with the advantage of biodegradability leaning more towards the PLA fenugreek scaffold as it lost more weight as time passed. 3. Mechanical properties. We tested the mechanical properties of both of the PLA scaffold and the PLA fenugreek scaffold in the tensile testing machine by subjecting it to pulling and stretching. The PLA fenugreek scaffold displayed a better mechanical strength compared to the PLA scaffold. 4. Cell migration. We took images of the cells before and after the scratch. Image A shows the cell prior to the scratch. Image B is after the scratch was made. The space in the picture is the scratch that represents injury. Image C shows PLA scaffold treated cell 48 hours after the scratch. Image D shows a sample treated by the PLA fenugreek after 48 hours. It was observed that the PLA fenugreek scaffold induced the most wound healing. As mentioned throughout my research, all the chemicals that have been used to manufacture this scaffold are of low cost. Therefore, I would imagine that the cost of
the overall cost of manufacturing the scaffold would be low. The chemicals used, like fenugreek and polylactic acid, ensure that the scaffold has anti-inflammatory properties and a hyperglycemic effect, which means it would be ideal for diabetic patients because it would lower their blood sugar levels and decrease the time needed for the wound healing process.
To conclude, PLA fenugreek scaffolds made by the electrospinning machine are suitable for tissue engineering and cell regeneration. The presence of fenugreek outside of the PLA provided a great water absorption property.
Thank you so much. This was Vanessa Ahmadi from Qatar University's Young Scientist Center.